Hello. Very good evening to the student who have joined. Can you let me know in the chat box if my voice is audible and if you can see the video and the presentation clearly? Okay, thank you so much. We will wait till more students join, or it is 6 p.m. and then we will start today's session. So very good evening to the students who have joined. We will wait till it's 6 p.m. and then we will start today's session. Meanwhile, let me know if you can hear my voice clearly or if there is any problem. I have already shared the presentation also. So if you have any problem regarding the presentation or the video or the audio, please let me know in the chat box. We will start shortly after 6 p.m. and so wait till then. Okay, so I think I will start with the introductions till other students join. It's almost 6 p.m. so let's start. So very good evening to the students who have joined. My name is Mousumi Deb and I am the PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL course of Medicinal Chemistry. Today it is our week 11 live session. So like every week, today we will have the content which is the week that is live right now, that is the week 12. So today it will be the last live session till we have the course left, and we will have 2 more live sessions again on Sunday on 14th April and another on Tuesday 16th April. That will be our last class. Okay, so in those last 2 sessions we will summarize the whole session, and I will give you questions from all over the sessions to see if you have remembered the things we have learned earlier in our first couple of weeks or not. And today it is our week 11 live session. So right now the week 12 course content is live. Okay, so today we will discuss the content which are in your week 12 course. Okay, so the topics which are involved in the week 12, today we will discuss those questions. So like every week what we will do is we will discuss around 20 MCQs. Each and every MCQ will have 4 options, okay, A, B, C and D, and you have to tell me in the chat box which of the following you think is the correct one, and then after some time I will give you 3-4 minutes, and after 3-4 minutes I will check the chat box, look at your answers, and then I will tell you which one of the following is the correct one, and then we will discuss the topics which like we do like every day. Okay, so today it is our week 11 live session. So before starting the session, what I do is, like on every week I summarize few things before starting the sessions. Who have attended my sessions earlier, you know how we do it. Like since I am recording the session, I have kept your microphones and video kept your microphones mute, and so that I can smoothly record the session. Okay, but if you have any doubt, any query, the chat box is open always. You can let me know at any time. Okay, since I am recording the session, so that there is a smooth streaming, that's why I have done it. So before starting, I will just summarize few things. Like there will be a problem solving session every week. Like today it is our 11th problem solving session, and we will have 2 more problem solving session. One is on 14th April Sunday from 11 am. That will be only 1 hour of problem solving session. That more kind of a summary session where I will summarize the topics we have learned earlier. And on the last our problem solving session will be on next Tuesday, that is on 16th April from 6 p.m. to 8 p.m. as 2 hour as usual class. In the last two classes, not only I will summarize the topics which we have learned so far, I will introduce you with few more important topics of medicinal chemistry that were not so much discussed in your course, but that are very important. And also I will give you some questions, okay, like question as in I will give you few questions like in earlier couple of weeks to see if you have remembered those things or not. This will help you to revision your things, right. So, the problem solving sessions are on every Tuesday from 6 p.m. to 8 p.m. These sessions will be live on Google Meet platform, and the Google Meet joining link for the sessions is same, which is the link you have used to join the class today. This Google Meet link will be same until we finish our course. So, if any network disruption happens, it is a very important point. If any network disruption happen from my side, do not leave the session, okay. I will rejoin as soon as possible, but I will request all of you not to leave the session. And if there is any network issue from your side, you can anytime you can just leave and rejoin, okay. So, there is no problem, you can rejoin at any time if there is any unstable network from your side. And since I am recording the session, and also I will share this recording and the PPT with you, so you do not have to worry if you have missed anything due to the poor network. During the sessions, the students can post their doubt, their question through the chat box, okay. If you have any doubt or any query, it may be from today's class or any previous sessions or any previous course topic, just let me know in the chat box, okay. I will happily answer your question. Even if I cannot do it today, I will do it in the last classes definitely. And if you any doubt persist after class, it may be like this, you find that there is any problem after you finish the session, you can email me. I will share my email ID at the end of the session, like every day, okay. You can email me any problem, any topic you think I need to repeat, you can just let me know, and I will request you to do it by 2 to 3 days next days, because I have to prepare along with, because in the last Tuesday we will have the last class, right. And this presentation class recording, as I have told you earlier, will be shared to you, okay. So, now let me start with today's session. Just let me check how many of you have joined till now. Okay. So, I hope my voice is audible to you. If not, just let me know. So, today it is our week 11 live session. Right now we have week 12 that is ongoing. This is the last week of this course content, right. This is the last week course content, that is the week 12. We have QSAR, and then it is divided into 1, 2 and 3 part, and then we have drug resistant and synergy, okay. So, like every week I will show you questions. This will be MCQs having 4 options. Any one of the options will be the correct one, and you have to identify what will be the correct option, okay. And before starting, I will just want to clarify, I just want to note down one more thing, that there will be a problem solving session. Just let me type it in the chat box, so that whoever joins, that there will be a summary session. So, the summary session is an additional 1 hour session on 14th April, that is on Sunday, from 11 p.m. from 11 a.m. to 12 p.m. On this summary session, what I will do is I will just pin it up, so that, yeah. So, this session will be for summary session. I will summarize the topics which are in the course, okay. The Google Meet joining link will be same, this link which you are using right now to join the session. You can join that on the same link to access the class. I request all of you to attend that class, okay. That will be a little bit important, because I will discuss the topics which were a little bit in your course, but that need a very elaborate demonstration. I will discuss these things. Also, I will try to revise you the things you have might forget. So, please join the session. I have just pinned it up. So, just note it down so that you do not forget. Maybe you will get a reminder email too, but from my side I just thought to share it to you, okay. So, now I will start with today's session. So, the first problem in today's session, it says that which of the following is not a QSAR parameter. Your options are number A, hydrophobicity, number B, hydrophilicity, number C, steric, and number D, electronic. So, which of the following is not a QSAR parameter? like every day, again I will request all of you, since these are almost our end, toward our end sessions. So right now I want all of you to participate and answer, okay, because your exams are nearer, because I want to know if you are following these things or not. Hello. Okay, there are answers. Okay, so already I am getting answers. Okay, I will wait for some few more time. I will wait for some time more. After 2 to 3 minutes, I will show you what is the correct answer, and then we will discuss what is QSAR, what are the parameters required from QSAR and everything. So let me check now the chat box. All of you, please answer the question, okay? Let me check the chat box. I think already many of you have answered the questions. Okay. So yeah, I have quite a few answers, and almost all of you, okay, all of you think, whoever answered, that it is the number B. Okay, now let me check if the number B is the correct one or not. Okay, and then we will discuss what is QSAR. I will discuss in detail. So the parameter which is not a QSAR parameter is, yes, number B, hydrophilicity. Hydrophilicity is not a QSAR parameter. So what is QSAR? So first thing which we have to know, what is a QSAR. So SAR we already have discussed earlier also, that SAR is a structure activity relationship. So what is a structure activity relationship? So basically it is the what is the different part of the compound. Let's say we have a compound, or if we have a structure. So there are various structural there are various part of the particular structure of a drug. Let's say some part have some role in hydrogen bonding interaction, some part of that structure have a role in the pi stacking interaction or hydrophobic interaction, and so on. So that is called the structure activity relationship. But a QSAR, it's known as a quantitative structure activity relationship. It is a mathematical relationship. The SAR we have discussed so far is a qualitative. We can say it is a qualitative one. There were not mathematical question or any parameter or any value. Okay, but QSAR, it is a very important part in case of the computational chemistry, also for computational biology, the in silico studies. Okay, for in silico studies, QSAR is a very important parameter. So quantitative structure activity relationship is a mathematical relationship which correlates measurable and calculable molecular properties to some specific biological activity in terms of an equation. QSAR attempts to find consistent relationship between biological activity and molecular properties so that these rules can be used to evaluate the activity of new compounds. Understanding QSAR regarding the electronic effect, steric effect and lipophilicity. Okay, so various physico-chemical parameters are there in QSAR studies. The important ones are hydrophobicity. This hydrophobicity is measured by partition coefficient, pi substituent constant. The steric parameter, that is the Tufts constant, Verloop steric parameter, electronic parameter like Hammett's constant and dipole moment. So these are very important physico-chemical parameter for QSAR studies, but hydrophilicity is not a QSAR parameter. Okay, so remember what are the QSAR parameters, that are the hydrophobicity, steric parameter and electronic parameter. Okay, so number B was the correct one. So now I will move toward the next problem. Problem number 2 says the role of efflux pumps in the drug resistant is due to, number A, metabolizing the drug before reaching the active site, number B, inactivation of drug, number C, decreasing the drug concentration inside cell, and number D, none of the above. So which of the following is the correct role of efflux pump in drug resistance? Again I will give you some time. I will wait for 3 to 4 minutes now, but I want all of you to answer. whoever are present in the class, read the options carefully. I know you can answer the question. Even if your answer is wrong, it doesn't matter. But I want all of you to try to answer the question. Okay, what is the role of efflux pump in drug resistance? The role of efflux pumps in drug resistance is due to number A, metabolizing the drug before reaching the active site, number B, inactivation of drug, number C, decreasing the drug concentration inside cell, number D, none of the above. All of you try to answer the question. I want all of you, whoever is present in the class, all of you should answer the question. Okay, let me check the chat box now. Okay, I think I have many answers. Number A, number B, number C, number C, number. Okay, so now I have multiple answers. It's like A, B, and C. Some of you think it is A, some of you think it is B, and some of you think it is C. Okay, let me check if which of the following is the correct one. So the correct answer will be the role of efflux pumps in drug resistance is due to number C, decreasing the drug concentration inside cell. So efflux pumps, these have very important clinical relevance. Okay, for some bacteria and eukaryotes, efflux pump can counter the MDR resistance. MDR means multi-drug resistance, and they can add themselves to other resistance mechanisms. So what are efflux pumps? Efflux pump can be, so because of the importance of efflux pumps are becoming a very important target in case of, especially in case of antibacterial drug discovery. Okay, so like for efflux pump inhibitor, by blocking the pump and restore the drug activity. So what are the efflux pumps? So efflux pumps are basically, they eject the antibiotic. So there is a bacteria, you can see, and these green portions, you can say it is your efflux pump. So whatever have to do, since we are ejecting an antibiotic to the bacteria. Okay, sometimes few amount of antibiotic is able to penetrate the bacteria, but some fails to penetrate the membrane, bacterial membrane. Okay, but efflux pump, what they do, whatever the antibiotics are entering, the efflux pump, they eject that antibiotic. So because of that ejection of antibiotic, what happens, the drug concentration inside the bacterial cell, it is decreasing, and because of that, the efflux pump pump, the bacterial cell became a drug resistance. So, inhibiting this efflux pumps are one of the very emerging and very new target in case of the antibacterial drug resistance, drug discovery. So, altered drug distribution to the active site of action is a very important topic. Efflux pump are the transmembrane protein that are often highly expressed by microorganisms and tumor cells that act as a transporters by binding to drugs and carrying them out of the cell before they can exert their therapeutic effect. Some transporters known as multidrug resistant pump are quite broad in their specificity and can efflux a variety of natural and synthetic drugs. Both synthetic and broad specificity efflux transporter systems have been identified that contribute to bacterial resistance to a broad range of antibiotic drugs. Okay. So, the role of efflux pump in drug resistance is due to the decreasing the drug concentration inside the cell. They pump out the antibiotics which are able to penetrate to the bacterial cell membrane, and because before the antibiotic can show its antibacterial effect, it actually ejects the antibiotic out of the cell. Okay. And remember, these are the transporters and multidrug resistant pump and quite broad in their specificity. So, these topics are very important, like you can have any kind of short note on this kind of topics. So, that is why this kind of topics are very important. So, read about the efflux pump very carefully. Okay. Now, I will move toward our next problem. So, problem number 3, it says that which term is used to describe the inhibition of two different enzymes in the same biosynthetic pathway. Your options are number A, parallel inhibition, number B, sequential blocking, number C, MT therapy, and number D, enzyme inhibition. Which term is used to describe the inhibition of two different enzymes in the same biosynthetic pathway? Number A, parallel inhibition, number B, sequential blocking, number C, MT therapy, and number D, enzyme inhibition. Again, read the options carefully and let me know which of the following you think is the correct one. Even if it is wrong, it doesn't matter because we are not giving a marks, but answering is very important. Otherwise, I will not know if you are understanding or if you are following the class or not. Okay. So, take some time, think and let me know in the chat box. Let me check the chat box now. Okay, so I have few answers. Like it said, it is number B, sequential blocking, number B, number B, number B. So I think all of you think it is number B. Still not all of you are answering the questions. Okay, from next question I want all of you to answer the questions. Let me check if sequential blocking is the correct answer or not. So the correct answer, it will be yes, it is the sequential blocking. So what is sequential blocking? So it is the inhibition of two or more consecutive steps in a metabolic pathway. The reason this is effective because it is difficult, particularly with a reversible inhibitor, to inhibit an enzyme 100%. If less than 100% of the enzyme activity is blocked, the metabolic pathway has not shut down. Okay, so what happens? Let's say we have a metabolic pathway like from which is like A goes to B, goes to C, goes to D, and goes to E. So let's say we have a particular inhibitor. Let's say we have a particular reversible inhibitor. So what happened in case of the reversible inhibitor? Can any one of you can tell me what happens because of a reversible inhibitor, or which inhibitor should we term as reversible inhibitor? We have different types of inhibitor, right? We have reversible, we have irreversible inhibitor, we have suicide inhibitor. So what is a reversible inhibitor? Let's say if you have remember it or not. Your examinations are almost round the corner, right? So what is a reversible inhibitor? Let me know in the chat box. I will wait for some time. What is a reversible inhibitor? Any one of you? Yeah, reversible or competitive inhibitor. So what is a reversible or competitive inhibitor? I will wait. Let me know in the chat box. I want to know if you remember this or not. Okay, I will just repeat that particular sequential blocking, but after that I will check if any of you can say. Yeah, which binds in place of substrate. Very good. So a reversible inhibitor is an inhibitor that binds mainly in case of in the active site, right? The active site of an enzyme where the substrate binds. But reversible inhibitor is bind to the particular active site. But we all know that what happens in case of a enzyme catalysis. What happens? Like the substrate is at a very high amount, right? So it is not possible for all inhibitor molecules to go and bind to the active site of the particular enzyme, right? So what happens in case of all these things I will repeat in the last summary class also. Okay, I will summarize these things, but I just wanted to check if you remember or not. So what happens in case of sequential blocking? So let's say we have this metabolic pathway where A goes to B, goes to C, goes to D, and then it goes to E, and E is our final metabolic pathway, which is our final product. This pathway we want to shut down. Okay, so let's say we have an inhibitor which particularly inhibits this particular step of B to C, but it is not possible to inhibit an enzyme 100% with a particular on enzyme. So if let's say we have inhibited that particular step up to 60%, but still the 40% is going on, and the metabolic pathway it not shut down yet, right? So it is not so it won't be very fruitful. But so what happen in case of sequential blocking? If let's say if we have another inhibitor, let's say we have another inhibitor which binds, one second, where is the pen? So let's say we have another inhibitor which inhibits this pathway also. So by this way, what can we do is we can inhibit that the total metabolic pathway a little bit more, right? Because let's say from this to this the 60% of the inhibition is happening, but this to this another 40% which are the remaining, it can happen or almost it can be stopped, the metabolic state pathway. Okay. So here it is written that let us say A goes to B, goes to C, goes to D and goes to A. So this is the metabolic pathway we want to inhibit. Now let us say we develop an inhibitor for this step. Okay. So here because D does not get converted into the C by inhibition, you do not get the formation of E, right, but less than 100% of this, of course we are looking reversible inhibition. It is possible you could get never 100% inhibition, right. So if you have less than 100% of the enzyme activity blocked, then the metabolic pathway has not really truly shut down. There is also going on some amount of E which is produced. But when you use inhibitors of two consecutive enzymes in pathway, so here we will say this A going to B, going to C, going to D, going to E. So what should we do? We would not have this block this, but perhaps you have to block this. Okay. So we will develop two different inhibitors, and this is also they think we can work in synergy. So this is the sequential blocking. We have to develop two different enzymes inhibitor and for a particular metabolic pathway, so that the metabolic pathway can be shut down completely, and this is known as the sequential blocking. And the inhibitors which we have developed, the two different inhibitors, they must work in synergy. The working synergy mean they should not they should not block each other's work. Okay. They should help each other. There should be a synergy in their action, mechanism of action. Okay. So that is the concept of the sequential blocking. Now I will go to the next topic. So the problem number 4 says that contour maps are present in number A 1D QSAR, number B 2D QSAR, number C 3D QSAR and number D all of the three. Contour maps are present in which QSAR? 1D, 2D, 3D or all of the three have this contour maps present. Contour maps are present in which type of QSAR? 1D, 2D, 3D or all of the three of them have this contour maps present? Again I am saying in the last, today is the last class that we are finishing our course today, right? We don't have any more topics left in the course. So the last two classes, one will be on next Tuesday from 6 PM to 8 PM, that will be our last class. But before that, on 14th April Sunday from 11 AM I will have a 1 hour summary class where I will summarize few topics of the course and also we will revise few things. So if you have any topic you think I need to repeat, or you didn't understand earlier, or you have any problem, so please let me know. Okay, in today's class you can let me know, or also I will share my email ID. I will expect in next 2 days you let me know what the topics you want me to repeat, if there is any topic or any problem, but please let me know before Friday so that I can prepare accordingly. Now let me check the chat box. Number B, number C, number D, number D. So I have one number B, one number C and multiple number D. Okay, let me check which of the following is the correct one. So the contour maps are present in number C, 3D QSAR. So what are contour maps? So basically in case of 3D QSAR, what happens, a set of active and inactive compounds are required from a congeneric series of compounds. The compounds are divided into training set and test set. The training set compound are used to build a 3D model. The test set compound are used to evaluate to and refine the model. So, in the 3D QSR consists of contour map indicating the favorable and unfavorable region. So, this is an example of 3D QSR. The green portion of the they are known as statically flavored, the yellow are statically non-favored here, and the red are electron rich and the blue on the electron deficient. So, basically in case of QSR, what happens, like this is a computational technique, right. So, let us say we have 100 number of let us say 100 number of any particular antibacterial drugs. Okay. So, what will happen, what will QSR will do, it will build a particular mathematical model by using the compounds which are already active and inactive, for in case of active or inactive. So, few compounds will be active, few compounds will be not active, and the model will be built using, let us say 80% of the compounds will be used to build the particular model. Okay. These compounds are known as the training set, and then we have a test set which will use to evaluate if our model is accurate or not, or if there is any problem or not. Okay. And this 3D QSAR model, they consist of contour maps. Okay. So, this is basically, if you can see, look at it here carefully, we have a particular compound, you can see, if you can see. If you remember in last class also I have shown you when I was discussing about the, I think, kinase inhibitors and the proteins also, I have shown you that how the kinase enzyme looks, how the 3D model looks. Okay. So, there is a techniques known as molecular docking and all these things. So, in this computational technique, the proteins can be visualized as a ball and stick or ribbon, and any I mean the molecular, the contour maps, all these things are used in this case. Okay. And the compounds, these can also visualize like this, and there are regions which can be indicate like these are the statically favored, or we can change the we can do modification in that position, or this straight press or electron rich or electron deficient, and so on. Okay. So, basically 3D QSR is the technique where the contour maps are present. So, now I will go to the next problem. Problem number 5 says, which of the following resistant mechanism describes the function of beta lactamase? Your options are number A, flux pump, number B, target mimicry, number C, drug inactivation, number D, target overproduction. Beta lactamase, we all know, is a very important enzyme, right, in case of this antibacterial drug resistance. So, what is the resistant mechanism that describes the function of this beta lactamase? Efflux pump, target mimicry, drug inactivation, or target overproduction. All of you try to answer the question. I will give you 3 to 4 minutes again, then I will check your answers, and then I will show and we will discuss it. Which of the following resistant mechanism describes the function of beta lactamase? Okay, let me check the chat box now. I think then we can discuss it. Okay, number C. So I have most of the cases. In case I have number C and few number A also. Okay, let me check. Number C is the correct one. Yes, drug inactivation is the main mechanism of resistance. Okay, resistance mechanism that describes the function of the beta lactamase. So there is a type of resistance called drug inactivation. Some bacteria can use these different methods to render an antibiotic useless, essentially by destroying the antibiotic structure. Okay, as you can see, here is the structure of an antibiotic, which is the penicillins, the basic structure of penicillins, which have a beta lactam ring. The beta lactamase, what does it do? It just hydrolyzes this beta lactam ring, which is essential for the activity of beta lactamase beta lactam antibiotics. Okay, and this is known as the drug inactivations. So here there is a mechanism how this beta lactamase hydrolyzes the this 4-member ring. This mechanism is also very important. Okay, so this is the serine beta lactamase, and we have two type of beta lactamase mainly. One is serine beta lactamase and one is metallo-beta lactamase. In case of serine beta lactamase, in the active side we have a serine, we have a glutamic acid residue, and we have a lysine residue. The glutamate residue and lysine residue, they act as a base, and the serine, it has a hydroxyl group, okay, which acts as a nucleophile. This nucleophilic this first attacks the carbonyl carbon of this beta lactam ring, and then we have this step is the acylation step, and there is a water in the vicinity of that particular active site. So what happened, this glutamate residue takes up the water and makes this water a more effective nucleophile, which is a hydroxide ion, and this hydroxide attacks to this position, and this is the deacylation. And because of this attack, we have this, now the lactam ring is opened, we have a carboxylic acid and an amine group here, and we have our inhibitor, the particular enzyme as it is. Okay, there which so this is the acylation step, and this is a beta lactamase action. Okay, then we have a metallo-beta lactamase, where the nucleophile is the hydroxyl group, which is attached to a zinc ion. This zinc ion is a 4 coordinate. We have 3 histidine units, and this zinc is in +2 state. This hydroxyl bound to zinc attacks to the particular carbonyl carbon, and the mechanism is similar as like before. Okay, this is the action of metallo-beta lactamase. So the resistant mechanism describes the function of beta lactamase is the drug inactivation. So now I will move toward the next problem. Problem number 6 says the sigma m value of the phenol substituent OH is 0.12, whereas the sigma p value is 0.37. What do this sigma m and sigma p they stand for? Number A, the electronic effect of phenol substituent at the meta and para positions respectively. Number B, the hydrophobic effect of phenol substituent at meta and para position respectively. Number C, the steric effect of phenol substituent at meta and para position respectively. And number D, the acidic effect of phenol substituent at meta and para position respectively. What does this constant stand for? Read all the options and question carefully, and then let me know in the chat box what does this constant stand for. Electronic effect, hydrophobic effect, steric effect, or acidic effect of phenol substituent at meta and para positions respectively. All of you try to answer the question. Okay, let me check the chat box now. Number A, number A, number A, number C, number A. Okay, I have number A, number C as different options. Okay, let me check which of the following will be the correct one, number A or number C. Okay, this is the correct answer will be number A, the electronic effect of phenol substituent at meta and para positions respectively. So, this sigma which I haven't told you yet, this is known as the Hammett substituent constant. Okay. So, the substituent constant is a polar reactions are strongly affected by substituent on the equilibrium constant and the rates of the reaction of organic molecules and ion. So, in 1937, Louis P. Hammett suggested that the effects of the meta and para substituent on ionization constant of benzoic acids could be the predictors of the electronic influences of substituents on variety of reaction. But why? Because a large number of substrate benzoic acids are readily available. Number 2, ionization constant are easily determined, and number 3, substituents on aromatic rings are held at a fixed distance from the points of the reaction. Okay. So, this sigma, it is also known as the Hammett substituent constant, which stands for the electronic effect of phenol substituent. Okay, not phenol substituent, and the electronic effects. Okay, and the particular what is the particular meta and para ionization constant of benzoic acids are taken as a reference for prediction of this particular electronic influences. Okay. So, now I will go to the next problem. Problem number 7 says which of the following is an inhibitor of inosine 5' monophosphate dehydrogenase. Your options are number A, dopamine, number B, misrobin, number C, propranolol, and number D, ciprofloxacin. Which of the following is an inhibitor of inosine 5' monophosphate dehydrogenase? A, B, C or D, which of the following will be the correct answer? Which of the following is an inhibitor of inosine 5' monophosphate dehydrogenase? Let me check the chat box. Number B, number B, number B, number B. So, all of you think it is the number B. Okay, let me check if number B is the correct one. Okay, so the correct answer is yes, very good. It is number B, misrobin. Okay, so this is the structure of misrobin. Now, can you tell me what does this ring called? This nitrogen containing heterocyclic ring. What is the name of this ring? This one. What is the name of this particular ring? Let me check the chat box. What is the name of this particular nitrogen containing heterocyclic ring? Imidazole. Yes, very good. So, when you are just reading the medicinal chemistry, now you have completed all 12 weeks. So, identifying the heterocyclic rings, especially the nitrogen containing heterocyclic rings, are very important, okay, because the questions can be asked on this matter also, about the structure of the drugs, okay. So, because of that you have to remember and you have to know what are the names of the particular heterocyclic containing nitrogen containing heterocycles, mainly nitrogen containing heterocycles. So, this is a structure of mesoribin. So, basically it is used in case of various renal diseases, okay, and this is an immunosuppressive drug, which you can see here. We have a particular this sugar phosphate sugar unit, okay, like this. So, this is metabolic pathway of guanine, dehydrogen and mesoribin. So, what does it do basically? So, we have this mesoribin and this it is converted to microfuronic acid mesoribin monophosphate, okay, by in the help of the adenosine kinase. And what does it do? There is a inosine monophosphate, and this inosine monophosphate is converted to RGMP, and which is goes to the particular this metabolic pathway to RNA, okay, and this the enzyme which helps in this pathway is the inosine 5' monophosphate, okay, dehydrogenase. So, this is which also known as the IMP dehydrogenase. So, particular mesoribin is an inhibitor of this dehydrogenase, and also we have this guanine PRPP which converted to RGMP and this way, and we have the ribonucleotide reductase also, okay, and this is mainly the guanosine pathway of DNA and RNA, and this drug is also used in case of the various the renal diseases, and it is a immunosuppressive drug. You do not need to memorize all this mechanism pathway, okay, all this metabolic pathway you do not need to remember, just remember the enzymes which is an inhibitor and the diseases all it is used, and also the structure. This up to this, if you remember, it is fine, okay. Now, I will go to the next problem. So, problem number 8 says, which of the following statements is true? Number A, hydrophilic compounds have a high value of P. Number B, hydrophobic compounds have a high value of P. Number C, acidic compounds have a high value of P. And number D, basic compounds have a high value of P. Which of the following statement is true? And also with the option, I want to know what is P. What do you mean by P? I haven't mentioned it yet, but I know many of you I think know what is P. I already have mentioned it, I think in previous third or fourth lecture itself. So, what is P and which of the following options is the correct one? A, B, C or D? If you know what is P, you can clearly answer the question very easily. So, which of the following statement is true? Hydrophilic compounds have a high value of P, hydrophobic compounds have a high value of P, acidic compounds have a high value of P, and basic compounds have a high value of P. What is P and which of the following statement is true? All of you try to answer what is P and which of the following statement is true. Okay, let me check the chat box now. Okay, P is a partition coefficient, ratio of drug distribution in organic and aqueous ratio. Okay, so I have many options. All of you think it is the number B, and also I have answer what is a P. Let me check if it is correct or not. So the correct option is yes, number B. Hydrophobic compounds have a high value of P. So P is a partition coefficient or distribution coefficient, which is indicated as D, is the ratio of concentration of a compound in a mixture of two immiscible phases at equilibrium. The most common physiochemical descriptor is that the molecule's partition coefficient in an octanol and water system. After administration of a drug will go through the series of partition steps, leaving the aqueous extracellular fluid, passing through the lipid membrane, entering the other aqueous environment before reaching the receptor. In this sense, a drug is undergoing the same partitioning phenomenon that happens to any chemical in separating funnel containing water and non-polar solvents such as hexane, chloroform and ether. So partition coefficient P or distribution coefficient D is defined as the ratio of concentration of a compound in the mixture of two immiscible phases at equilibrium. The biphase liquid phase, specifically ionized solutes, or a logarithm of is known as log P. Hence partition coefficient measures how hydrophobic or hydrophilic a chemical substance is B. Okay, if the value of P is very high, the compound is known as to be the hydrophobic one, and if the value is low, it known as the hydrophilic one, and this is known as the separating funnel. Okay, so in chemical lab, what happens whenever you are having a reaction, sometimes what happens, like the after the reaction we have to do the workup of that reaction. So that is what is the workup, or it is also known as the extraction process. So what does it do? So basically, if there is any metal, especially if there is any metal kind of reaction or any kind of salt formation which are not our desired product, so what we do is we take that reaction mixture in a separating funnel. This is known as a separating funnel. We had water in it, and also we have to add a particular organic layer. Sometimes it is a chloroform, sometimes it is a non-polar solvent like DCM, ethyl acetate on those, and then we have to take it in this separating funnel and we have to shake it. So if we shake it, if the based on the density of that particular non-polar solvent, okay, there are two phases. In case of dichloromethane or DCM and water, the DCM is heavier one, so it stays in the lower portion and water goes to the upper portion. In case of water or ethyl acetate, in case the water is a heavier one, so it stays in the lower portion and the organic layer goes to the upper portion. So organic substance, mainly the product, starting material and whatever these are, they goes to the organic portion, and the salts and any catalyst and anything which have a hydrophilic ones, so they goes to the water layer. Similarly, in case of the drugs, so also they have the more the value of P, more is the hydrophobic is that particular drug. Sometimes we need the drugs which are the hydrophilic ones, and sometimes we need the drug which is the hydrophobic one. That's how this P is a very important function or a very important criteria in case of the drug discovery, and also in case of the activity of the drugs. Okay. And if you remember, we have this log P value in case of Lipinski's rule also, which gives the drug likeliness of a compound. Okay. So now I will go to the next problem. So problem number 9 says aminoglycosides resistance is due to number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of an electrical potential gradient across the bacterial membrane. Aminoglycoside resistance is due to which? Number A, target mimicry, number B, target overproduction, number C, efflux pump, and number D, lack of an electrical potential gradient across the bacterial membrane. Again, all of you try and answer the question. After 2 to 3 minutes, I will check and I will tell you which of the following is the correct one, and we will discuss it. Should I check the chat box now? Okay, let me check it. Okay, number D, number D, number D, number B, number B. So I have few number B and few number D. Okay, let me check which of the following is the correct one. So the correct answer will be, yes, it is number D, lack of an electrical potential gradient across the bacterial membrane. So aminoglycoside antibiotic resistance can arise from the lack of quinones that mediate the drug transport, or from lack of an electrical potential gradient required to drive the drug across the bacterial membrane. So aminoglycoside, it is a medicinal or bacteriology category of traditional gram-negative antibacterial therapeutic agents that inhibit protein synthesis and contain as a portion of the molecule an amino-modified glycoside, which is sugar. Aminoglycoside antibiotics display bactericidal activity against gram-negative aerobes and some anaerobic bacilli that resistance has not yet arisen, but generally not against gram-positive and anaerobic gram-negative bacteria. So one of the ways in which that can occur is by changing the net charge on the membrane. So if you can for example, if there is a phosphatidylglycerol, membrane can be changed to L-lysyl phosphatidylglycerol groups and converts to aminic group to cationic group. So this change can affect the permeability of a drug. So if the permeability of the drug goes down, clearly its efficacy will go down. In this way, the drug with some change can actually now be repelled. So aminoglycoside antibiotic, for example, became inactive because the quinones that mediate the drug transport, or from lack of the quinone that mediate the drug transport, or from the lack of an electric potential gradient required to drive the drug across the bacterial membrane. So these two can result in decreased uptake of aminoglycosides, which are going to result in drug resistance. Okay. So also, not only the lack of electrical potential gradient across the bacterial membrane is the reason for the aminoglycoside resistance. Okay, it can also arise from the lack of quinone that mediate the drug transport. So just remember it. Now I will go to the next problem. Problem number 10 says, which of the following is not the mechanism of resistance of quinolones? Your options are: number A, lack of potential gradient; number B, single point mutation; number C, active efflux system; and number D, mutation in the genes for DNA guidance. Which of the following is not a mechanism of resistance of quinolones? Number A, lack of potential gradient; number B, single point mutation; number C, active efflux system; number D, mutation in the genes for DNA guidance. me check the chat box. Number A, number A, number A, number C. I have most of the answers in number A and some number C. Okay, let me check if it is correct, which of the following is correct one. So the mechanism which is not a mechanism of resistance of quinolone is the number A, lack of potential gradient. So basically the bacterial resistance to quinolone resistance can emerge during the therapy, especially with S. aureus or S. aeruginosa. A single mutation is sufficient to cause resistance. Resistance is chromosomal rather than plasmid mediated. Mutation occur in the genes of DNA guidase or topoisomerase type 2, appear to be primary side of phenomena and other gram positive active efflux pump, which is present in both positive and negative. So active so mutation in the genes of DNA guidase, active efflux pump and single point mutation, all of these are mechanism of resistance of quinolone, but not the lack of potential gradient. Okay, so number A was the correct answer. Now I will go to the next problem. Problem number 11 says drug resistance of trimethoprim is generated due to number A, efflux pump, number B, sequential blocking, number C, mutation in target, and number D, insufficient drug concentration. Drug resistance of trimethoprim is generated due to efflux pump, sequential blocking, mutation in target, or insufficient drug concentration. Which of the following will be the correct one? Again I am saying, if you have any doubt or any query regarding any topic, please let me know. Okay, in today's class or after the class through the email, please let me know so that I can discuss those in the last two summary classes. Again I will give you some time, think and let me know, then after that I will discuss it. Let me check the chat box. Okay, number D, number C, number C, number B, number C. So I have multiple different options. I think number A, B, C and D. I think I have not A but number C, number B and number D. I have 3 different answers for this question. Okay, that is good. Now let me check which of the following is the correct one. So the drug resistance of trimethoprim is generated due to Number C, mutation in target, that is the reason for the drug resistance of trimethoprim. So, basically, so let us say you have a protein, so the mutation can occur here and this results in conformation change which changes the binding pocket for the enzyme. So, this is now not very good for the binding anymore. For example, resistance to antibiotic trimethoprim derives from a single mutation of a single amino acid of a target enzyme, which is dihydrofolate reductase. So, the properties of the singly mutated enzyme differ somewhat from the normal enzyme, but this is still binds to the substrate dihydrofolate. But now, the particular drug that cannot bind to the this antibiotic cannot act on the single the mutated substrate enzyme, which is the dihydrofolate reductase. So, basically, in what is the trimethoprim, that does it do? So, there is a pteridine and para-aminobenzoic acid which converted to dihydroteroic acid and which converted to dihydrofolic acid and tetrahydrofolic acid. So, this is the folic acid metabolism which is very important in case of the bacterial and also the for human life cycle also. So, in case of bacterial life cycle, what happens, like this conversion of pteridine or para-aminobenzoic acid to dihydroteroic acid is catalyzed by dihydrofolate synthesis, okay, and sulfonamides, they inhibit this particular step also. This is also an example of a sequential blocking. If you look at it carefully, from pteridine to para-aminobenzoic acid to tetrahydrofolic acid, in this particular total pathway, there are two different way we can just two different inhibitors we can actually use to completely block this mechanism. One is sulfonamides which inhibit the dihydrofolate synthesis, okay, which is required for this step, and then for the next step there is dihydrofolate synthesis which converts the dihydroteroic acid to dihydrofolic acid. Then we have dihydrofolate reductase which is inhibited by trimethoprim, which is which inhibits the conversion to the tetrahydrofolic acid. This is also one of a very good example for sequential blocking, because here two different inhibitors are blocking a particular metabolic pathway. Okay. So, remember, drug resistance of trimethoprim is generated because of the single point mutation of dihydrofolate reductase enzyme. Now, after the mutation, this now inhibitor molecule cannot bind with this enzyme, but the substrate still can. Okay. Now, the next question, problem number 12, it says that given the following log P values, what is the log P value for the di-substituted structure? So, these are the values are given, the log P values. For single benzene ring, the log P is 2.13. For for OME, this have log P, this particular 2.11. Then we have this one fluoro monosubstituted, we have this log P is 2.27, and then para OME and fluoro we have log P value. What is the log P value? You have to now identify it. Your options are number A 1.97, number B 2.01, number C 2.25 and number D 2.29. You have to identify the log P value of the particular di-substituted structure, this one, taking help of this one. This is a mathematical problem. I will give you a little more time for this, think and then do and let me know which of the following is the correct one. Let me check if I have any answers. Okay, I have many answers. Number C, number C, number D, number C, number C. I have few number C and few number D also. Let me check if number C is the correct one or not. So, the correct answer will be yes, number C. Very good. So, how to solve this kind of problem? So, basically log P will be the log P of benzene plus the pi of F and pi of OMe. Okay. So, how to calculate this one? So, this is basically the effect of the substituent. So, for calculating the effect for fluorine, what you have to do is you have to just subtract the log P value of the benzene, right. Okay, so this is the for the pi value of the particular fluorine is 2.27 minus 2.13, and the same for OMe is the 2.11 minus 2.13. It can be negative also. It will be positive in cases of your electron withdrawing ones, and for electron donating one it will be negative. So the log P value of this disubstituted one will be the base value, which is for benzene, and then you have to just add the two other values, which is 2.25. So very good, number C is the correct one. So now number 13, it says that when two drugs act on the same receptor, the resulting drug interaction is said to be which of the following? Number A, predictable, number B, unpredictable, number C, additive, and number D, empty therapy. When two drugs act on same receptor, the resulting drug interaction is said to be which of the following? Predictable, unpredictable, additive, or empty therapy. Okay, let me check. I think many of you can answer this question easily. Number B, number B, number D, number D. So I think number B and number D are the two given answer. Okay, I will give you one more minute if anybody else also wants to answer the question. When two drugs acts on the same receptor, the resulting drug interaction is said to be which of the following? I will give you one more minute. Let me check if you have few more answers. Number B, number B, number B. So I have number B and number D as the answers. Okay, let me check if which of the following is the correct one. So number C, the correct answer is number C, additive. So drug synergy, when drugs are given in combination, their effects can be antagonistic, subadditive, additive, or synergistic. Drug synergism arises when the therapeutic effect of two or more drugs used in combination is greater than the same effect of the drugs administered individually. So, you can think about the synergy by inhibiting drug destroying enzyme. So, we have already looked at this particular example. For example, you can have an inhibitor of enzyme that destroys the drug, and it will result in synergy. This particular molecule may not have the therapeutic effect on its own, but it only protects the drug for being destroyed. So, this is also called the sensitizing the drug or sensitizing the pathogen to the drug, but its primary target and inhibitor of destroying enzyme are administered together, then together they can have a synergistic effect. For already seen the previous in case of ampicillin and clavidumate, which is the example of the augmentin. I have discussed it in the last class itself, or the last to last class, I do not remember exactly, but I have discussed it, the ampicillin and clavidumate combination. So, they are administered together, and so that they can protect the ampicillin from beta lactamases. So, the topic of drug synergy, which of the part of the drug alliances, and we are going to look into that a bit more formally. So, drug synergy, when two drugs are given in combination that can be antagonistic, that means one drug is going to cancel the effect of other drug, or it can be subadditive, means there is going to be a little bit of addition of efficacy, or it can be additive, that means that A can be added to the effect of the B, which is going to result in a combination therapy. But more importantly, when we look at the synergy, then synergy refers to when therapeutic effect of two drugs are used in combination is greater than the sum of the drug administered individually. The difference between the additive and synergy is when I add these two drugs, the combination at which the efficacy is net effect does not go up, but in case of synergy, the sum of his effect is more than the individual drug. So, there is a difference between additive and synergy. Okay, additive means when I am giving a two drugs individually and the two drug in combination, the combination does not have higher efficacy. Okay, the efficacy do not go up, but when case of synergy, the combination of the drugs are the effect of the combination of drug will be higher than the individual drugs effect. So, that is the difference between additive effect and synergy effect, which I think many of you can get sometimes confused about. So, I have just added it here also. What is this drug synergy? Okay, so when two drugs acts on same receptor, it is known as the additive effect. Okay. Now, next problem number 14 says which of the following statements regarding the drug salmazole is false. Number A, the drug is free from CNS side effect. Number B, the log p value of the drug is 1.17. Number C, the 4 OMe group was replaced by 4 SOMe group, and number D, the log p value of the drug is 2.59. Which of the following statement regarding the drug salmazole is false? Which of the following statement regarding salmazole is false? Let me check the chat box now. Okay, number D, number C, number B, number D, number B, number B, B. So I have B, C and D, three different options. Alright, let me check which of the following will be the correct one. So the correct answer will be yes. The log P value of the drug is 2.59. This statement regarding the salbutamol is false. Okay. So altering log P value to remove central nervous system side effects. The cardiotonic agents, which is this one, okay, the cardiotonic agent was found to produce bright visions in some patients, which implied that it was entering the CNS or central nervous system. This was supported by the fact that log P value of the drug was 2.59. In order to prevent the drug entering to the CNS, the 4-OMe group was replaced by the 4-SOMe group. This particular group approximately have same side of the methoxy group, but it is more hydrophilic. The log P value of the new drug, which is known as the salmazole, was found to be 1.16. 1.17. The drug was now too hydrophilic to enter the CNS and was free from CNS side effect. So the statements which says first one, this drug is free from CNS side, which is true. The log P value of the drug is 1.17, it is also true. The 4-OMe group was replaced by 4-SOMe group, it is also true. But the last statement which says that log P value of the drug is 2.59, this statement is false. Okay. Initially the log P value of the drug was 2.59, means the drug was too hydrophobic. Because of the hydrophobicity of the drug was high, what was it doing? It was entering the CNS system and the patients were having the side effects of the drug because of the CNS effect. Okay. But then the reason of this particular hydrophilicity was the OMe group. So whenever we are having OMe here, the drug becomes very active and I mean very hydrophilic and enters to the CNS. But when we just change this OMe with the SOMe, so what does it do? This SOMe have a similar size, okay, of this OMe, but it is not so much hydrophilic. Because of that, the log P value decreases up to 1.17 and now the drug is too hydrophilic and it cannot enter the CNS. So this is a very good example how the SAR have been done. Okay, this is again a effect of SAR. Like we have a particular, we change the groups to see how the drugs are affecting the different particular properties of the drug is changing. So this is a very good example. So now problem number 15, it says that which of the following statement regarding the standard deviation is true. Your options are number A, standard deviation is the measure that is used to quantify the variation or dispersion of a data set. Number B, a low standard deviation indicates the data points are spread over a wide range of values. Number C, a high standard deviation indicates the data points are too close to the mean of the data set. Number D, none of the above. Which of the following statement regarding the standard deviation is true? Read all the options carefully, then let me know. Which of the following you think is the true statement regarding standard deviation? Okay, let me check the chat box now. Number D, number A, number A, number A, number. Okay, I have few number A and one number D. Let me check which of the following statement regarding the standard deviation is true. So the correct statement will be number A. Standard deviation is the measure that is used to quantify the variation or dispersion of a data set. So standard deviation, which is known as SD or S, is a measure that is used to quantify the amount of variation or dispersion of a data set. A low standard deviation indicates that the data points tend to close to the mean, also called the expected value of a set, while a high standard deviation means the data points are spread out over a wider range. Okay. Other statistical tests are also conducted to validate this trend. So this is a particular normal distribution curve, which is known as a normal distribution curve, and this is the calculating the standard deviation. This is the formula which is the summation of xi minus x bar divided by n minus 1. This is for the summation will be the whole square, where n is the number of data points, xi is the each of the data, and is x bar is the mean of the xi. Okay. So it is a statistic, that means that the dispersion of data set relative to its mean and is calculated as the square root of the variance also. So problem number 1, so statement number 1 was the true regarding the standard deviation. Now next problem, problem number 16 says that which of the following statement regarding the Craig plot is false. Your options are number A, it is a plot where x axis indicates the value of pi factor and y axis indicates the value of a sigma factor. Number B, this plot shows the close relationship between sigma and pi. Number C, from this plot it is possible to tell which substituent have a positive pi and sigma values, which substituent have a negative one. Number D, from this plot the substituent having similar pi values and similar sigma values can be observed. Which of the following statement regarding the Craig plot is false? Okay, let me check the chat box now. Number A, number D, number D, number D, number D. Okay, so number A, number D. I have two answers. Let me check which of the following will be the correct one. So, the correct answer will be number B, which says that the plot shows close relationship between pi and sigma. This statement is regarding Craig's plot is false. So, what is a Craig plot? Although the tables of pi and sigma factors are readily available for a large range of substituent, it is often easier to visualize the relative properties of different substituents by considering a plot where y axis is the value of pi and the sigma, and the x axis will be the value of pi factors, such as known as the Craig plot. The example is shown. I will show you the figure, which is this one, the Craig plot figure. This is the sigma and pi factor in para-aromatic substituents. There are several advantages to use of the Craig plot. The plot shows clearly that there is no overall relationship between pi and sigma. The various substituents are scattered around all the 4 quadrants of the plot. It is possible to tell at a glance which substituent have the positive pi and sigma parameters, which substituent have a negative pi and sigma parameter, and which substituent have one positive and one negative parameter. It is easy to see which substituent have similar pi values. For example, ethyl, bromo, trifluoromethyl, trifluoromethyl sulfonyl groups are all approximately on the same vertical line of the plot. In the theory, these groups could be interchangeable on drugs, whereas the principal factor affecting the biological activity is the pi factor. Similarly, groups which are from a horizontal line can be identified as being isoelectronic or having similar sigma value, like COOH, chlorine, bromine and iodine. Okay, so this is the Craig plot. So this plot shows do not show the close relationship between the pi and sigma. It shows that there is no overall correlation between these two values. Okay. So now I will go to the next problem. Problem number 17 says, which of the following statement regarding bioisostars are false? Your options are: number A, if sigma p is the most important physiological parameter for biological activity, then COMe would be bioisostar of SOMe group. If pi, number B, if pi is the most important physicochemical parameter for biological activity, then COMe, it would be the bioisostar of SOMe group. Number C, if pi is the most important physicochemical parameter of biological activity, then SO2Me will be bioisostar of SOMe. And number D, if sigma p is the most important parameter for biological activity, then SO2Me would be bioisostar of SOMe. Read all the options very carefully and then let me know which of the following statement is false. The statements are little bit tricky, but read all the options carefully. I know you can already figure out which of the following is the correct statement, I mean the correct answer. I will give you some time, think and let me know. Now let me check the chat box. Number C, number C, number A. Number C and number A. I have two different answers. Okay, let me check which of the following will be the correct one. So the correct answer will be both A and C are the correct one. Okay. So the table of substituent constant are available for various physicochemical properties, and knowledge of this constituent allows to medicinal chemists to identify substituents which may be potential bioisoster. Thus, the substituents of CN, nitro and COMe have similar hydrophobic, electronic and static factors and might be interchangeable. Such interchangeability was observed in the development of cimetidine and cimetidine analogues. That important thing is to note that the groups can be bioisostatic in some substituent but not others. Consider the example it shows that the physicochemical parameters for 6 different substituents. If the most important physicochemical property is sigma p, then the COMe group would be reasonable bioisoster for SOMe group, and however dormant parameter is pi, so then more suitable bioisoster of SOMe would be SO2Me. Problem number 18, it says that which of the following is not a property of drug destroying enzyme inhibitor? Number A, it only protects the drug from being destroyed. Number B, it has own therapeutic benefit if administered alone. Number C, if the primary drug and inhibitor are administered simultaneously, then it is more effective. And number D, none of the above. I think I have discussed this question indirectly. Which of the following is not property of drug destroying enzyme inhibitor? Number A, it only protects the drug from being destroyed. Number B, it has own therapeutic benefit if administered alone. Number C, if primary drug and inhibitor are administered simultaneously, then it is more effective. And number D, none of the above. Let me check the chat box now. Number C, number B, number B, number A, number B. So I think I have three different answers, A, B, C. I have three different answers for this question. Let me check which of the following is the correct one. So the correct answer will be number B. It has own therapeutic benefit if administered alone. This is not a property of drug destroying enzyme inhibitor. So inhibition of a drug destroying enzyme, this molecule has no real therapeutic effects of its own. It only protects the drug from being destroyed. Alone it has no therapeutic benefit. But if primary drug and inhibitor of a drug destroying enzyme are administered together, then the drug survives the destruction and is effective again. This is a synergistic effect. Okay, I already have discussed the synergistic effect. There I have told that if a drug and an inhibitor of a drug is administered simultaneously, it will give rise to a more effective combination, which is also known as the drug synergy. But this drug destroying enzymes, they do not have any therapeutic benefit of its own. Okay, it only protects the drug from being destroyed. But if administered alone, it do not have any therapeutic benefit. So the primary drug and inhibitor of a drug drug destroying enzyme, if they are added simultaneously, they will give rise to the synergy, which will have a more greater efficacy. Okay. So now I will go to the next problem. The problem number 19 says, what is the term coined when the therapeutic effect of two or more drugs used in combination is greater than the sum of effect of drug administered individually? I know all of you can answer this question. The options are number A antagonistic, number B sub-additive, number C additive, and number D synergistic. Which of the following is the correct one? I know all of you now can answer this question very correctly because I already have discussed it multiple times. Take some time and then let me know. Let me check the chat box now. Okay. Number D, number D, number D, number C, number D. Let me check. Yeah, number D is the correct one, the synergistic. Already I have discussed it lot of times, but again I will discuss it. So when the drugs are given in combination, their effects can be antagonistic, sub-additive, additive, and synergistic. Drug synergism arises when the therapeutic effect of two or more drugs used in combination is greater than the sum of the drugs administered individually. For example, the combined drugs action, the common example is the sulfur drug and trimethoprim. They are better as a bacterial killer. I have already discussed it. This is also a very good example of sequential blocking. Then we have aspirin and warfarin, which are the for hemorrhage. Okay, then we have captopril and nitro, which are the for blood control, the blood pressure control. So these are the example of synergism. And the last question of today's session, it says that what term is used to describe the process by which the best line are fitted through the set of data points of the graph. Number A, linear regression analysis by by the least square method. Number B, linear fitting analysis by the least square method. Number C, linear fitting assessment by the least square method. And number D, linear regression assessment by the least square method. Which of the following term is used to describe the process by which the best line is fitted through a set of data points on a graph? What term is used to describe the process by which the best line is fitted through a set of data points on a graph? Okay, let me check the chat box now. Number A. So, all of you think it is the number A. Okay, let me check if number A is the correct one or not. So, the correct answer will be number A. Linear regression analysis by least square method is the process by which a best line is fitted through the set of data points on a graph. So, a form of a mathematical regression analysis used to determine the line which is best fit from a set of data, providing visual demonstration of the relationship between the data points. The linear regression analysis or least square method is the common technique estimating the line of best fit. Once equation for a line is best fit has determined, forecast can be made. The least square method of linear regression involves the using the following formula for A and B, for y equal to a plus bx, where b is the n sum of xy minus sum of x sum of y divided by n sum of x square minus sum of x whole square, and a equal to y bar minus b of x bar. So, what are these terms? N is the number of data points on the theta, x bar is the mean of x value for all pair of data, and y is the mean of y value for all pair of data. So, we all have used this least square method and linear regression a lot. I think in statistics there are very mathematical equations. Okay. So, this was the last question of today's session. So, today it was our week 11 live session where we discussed the week 12 course content. So, we have successfully completed whole the medicinal chemistry course, the all 12 weeks we have discussed many questions, we have many classes and all, and now I will share my email ID here, okay, in the chat box. This is my email ID. So, since we have completed all the topics of medicinal chemistry successfully, now remaining to I have today it is the week 11 live session. We have one more live session left which will be on next Tuesday from 6 p.m. to 8 p.m. okay, as usual, and one more class, one additional 1 hour summary class I will take on Sunday. Okay, this Sunday 14th April from 11 a.m. to 12 p.m. 1 hour I will take a summary class. There I will summarize the topics which are important for medicinal chemistry but was not too much involved in your course, or some topics I will just revise, okay, and I will give you few questions, but mostly I will just give a lecture type of thing on in these last two classes which are remaining left. One is the summary one and one is our usual week 12 live session class. And apart from the topics I have chosen to get the revision. If you have any topics which you think I want to just repeat, or you have any doubt or any query, just you can let me know, okay. I have given my email ID, please let me know the topics which you need me to repeat or for your revision, so I will just prepare and I will let you know, okay. Okay, the recordings of the session, I will just show you how to find the recording of the session, okay. So, what you have to do is you have to go to the SWAM portal at first, okay. I will just show you, okay. Okay, let me just show you how to find the problem solving session recording. This one, just 1 second, yeah. So, first you have to go to the SWAM portal, okay. I think all of you have registered to the SWAM portal, right. For the classes you have to go to the SWAM portal at first. Let me show you how to find the problem solving session recording. I have few or others also have asked me it before. So, I will just show you. So, first you have to log in. Okay, after logging into the SWAM portal, you will have this kind of this particular kind of where you are finding the all your week 1, week 2, the lecture videos, also the assignment, all are here is present. In this portion, this left portion, in the course outline, you will have this problem solving session January 2024. If you click on that thing, so you will have this option problem solving session recording. Then if you click on that problem solving session recording, you will have a particular drop pop-up which says click here. If you click on that pop-up, so you will have this kind of sheet, Google sheet, okay. In this Google sheet you will find the link for the presentation, this is for the presentation link, and this is for the YouTube link for the videos. All the lecture videos I upload on the YouTube after the class, that same day or the next day I upload. So you will find there all the lecture videos, okay, and also you will find there is attendance and everything there, okay. So if you go to the particular this sheet, and then if you click on the YouTube lecture, you will find your particular lecture class recording and also the particular this thing, okay. Thank you all. I hope I could have helped you for the medicinal chemistry course, and since we have 2 more lectures left, so please join, okay. On the last 2 lectures, one will be on Sunday 14th April, you will find a reminder email also, so please join on a Sunday from 11 am and also on our last class on Tuesday. So please let me know in the email, okay, if you have any problem, any query, and you can also give a feedback, okay. So where you are finding the problem solving session, there is a thing called survey on problem solving session. If you click on that, you will find a particular Google form which is for the feedback for the problem solving session, okay. You can also give the feedback if the problem solving session was helpful to you. I ask all of you to give the feedback also for the same also, okay. So thank you all. I will close the session for today. If you have any doubt, any query, feel free to reach me through the email, okay. I will close the session for today. Let's meet on 14th April Sunday from 11 am for our summary session. So thank you all. I am closing the session for today.